Thank you everyone for joining our webinar today, Major IT Considerations for Genomics and Healthcare. We want to go over a few housekeeping items before we move forward with the presentation. Do want to let everyone know that the webinar is being recorded today, and there will be a Q&A section at the end of the presentation. Please feel free to add your questions to either the Q&A box or the chat box. And with that, Omar, take it away.
Good morning, good afternoon, everybody. I'm here to present the major IT considerations for genomics and healthcare, DNAnexus in partnership with Microsoft, and essentially how we're using NGS to optimize and de-risk clinical trials and to deliver clinical diagnostics. And essentially the molecular, clinical, and regulatory drivers that really make these considerations for IT. After we talk a little bit about how NGS, why NGS, we'll get into the DNAnexus Platform architecture that's been based upon these IT considerations, so you can understand how DNAnexus over the years has adapted and delivered to these requirements, particularly around the security and regulatory compliance. Following that, there will be just a very brief overview of DNAnexus, and then we'll go into a question and answer session. So why NGS in clinical trials and diagnostics? This will be our sojourn into the scientific side. A disclaimer, I am not a PhD. My experience is very deep in cloud and in setting up data centers, but for the last 5 years, I've had the privilege of working around some very, very smart scientists here at DNAnexus, and I've picked up a little bit of the science behind it. And it is fascinating. I would like to share just a little bit about that because the use of NGS in clinical trials and diagnostics is emerging, and I'd like to share why it's emerging, particularly in clinical trials. So the NGS opportunity is really about money. As you can imagine, the cost of drug development has just been going through the roof. At this point, it costs about $2.6 billion to bring a single drug to market. And the bulk of the expense is really due to failures, not successes, and these are late stage failures. Over 75% of the failures are in later stages, over 75% of the failures in the later stages are due to not meeting safety and efficacy objectives. NGS provides an opportunity to de-risk and optimize clinical trials using genomic biomarkers to stratify participants, to refine inclusion and exclusion criteria, and to track progress and impact of the clinical interventions. The key thing on the right-hand side here is to really note that from a phase 1 to approval, you have pretty much a 3 times higher likelihood of success if you have a biomarker. So biomarkers definitely figure very prominently into cutting into this $2.6 billion of cost for a single drug. The way we want to reduce that is to de-risk, which means we'd really like to know things sooner rather than later. If you get into a phase 3, you're talking a considerable amount of expense, and to come out of a phase 3 without the suitable efficacy results is really, it's a crying shame. The clinical trial solution that DNAnexus has recently launched incorporates these considerations, these IT considerations around molecular data handling, clinical data handling, and the regulatory overlay for all of that. For NGS in clinical trials, it's really about leaving no biomarker behind. There's a lot of data that's being left on the table, and our vision, and we've seen this vision echoed from our pharma customers, is that the world of NGS for everybody in the clinical trials is coming in the very near future. We want to be able to aggregate genomic information across studies and programs, so it's not enough to just take a given arm or a given study underneath a given phase of a program. We would really like to have a pan-study and pan-program view of the genomic information. There's a lot of reasons to do this. Larger cohort sizes will lead to better signal in relating genomic information to clinical information. And we would like to unify the NGS analysis and reporting across CROs and partners. one of the challenges that our pharma customers have had is working with different CROs, they get different results back. That's because the pipelines are not unified, the reporting's not unified. So that's another advantage of integrating providing a central IT solution, cloud-based IT solution for all research partners and CROs. This also enables us to maximize site and participant availability and to enable virtual trials. A virtual trial you can think of as almost like a siteless trial, and this has been gathering a lot of steam out in the world where intervention drugs are sent directly to patients, and this obviously will allow us to maximize participant availability and site availability. The discovery of new biomarkers and opportunities for basket trials and off-target applications are definitely enhanced by aggregating the genomic information across studies and multiple drug development programs, combining it with study, clinical, and real-world data. A secure cloud-based service unifies this analysis and reporting, as I said, across CROs. And the service that respects local data handling, again, enables us to go into sites and places and participants that might not otherwise have been able to be incorporated into this clinical trial. The discoveries are rapidly translated from research into regulated pipelines by having essentially a compliance continuum. This is another major IT consideration. You do not want to have little compliance islands of systems, some of which are validated, some of which are not. There is a need for change control, for versioning, for all of these things throughout the entire research and clinical development process. So it is not efficient to be able to have to pick up from one regulatory domain, say a research domain, and then go through all the heavy lifting of hardening and validating pipelines into a clinical compliant domain. We would also like to be able to create genetically mapped pre-screened participant cohorts, having at our fingertips a set of cohorts for a variety of therapeutic areas that are already genetically mapped could be very valuable in reducing the time to set up and execute a clinical trial, and to be able to design a clinical trial much more rapidly with knowledge, foreknowledge of various relationships between genome condition, genome state, and clinical state. The utility of NGS biomarkers in clinical trials will be demonstrated in 2 therapeutic areas. This is really the driving force behind incorporating genomics into both clinical trials and to diagnostics. The line is definitely blurring from R&D and discovery into clinical trials. We need specialized workflows. We need central data retrieval and production. We need real-time analytics within the clinical trial container, as it were, the regulatory boundary. There are rapidly evolving analytic tools for R&D that need to be deployed. Again, we need to be able to move and translate research discoveries rapidly into clinical diagnostics and clinical trial. The FDA environment for genomic analysis is evolving, and we need to stay on top of that. And we need an increased focus on clinical trials as a rich sample set. One of the examples of this is the AstraZeneca 2 million patient cohort that they're embarking upon to relate clinical data to genomic state for this large cohort. Use of clinical grade NGS is required to determine if a drug is working in an efficacy readout. Similarly, for a safety readout, we need clinical study grade NGS to determine if we have any off-target undesirable genomic effects and to carry out post-market surveillance. In oncology specifically, as the genomic makeup of tumors is becoming more and more smaller subsets, we're seeing we have to be much more selective in our patients for a study based on their genetic profile. It doesn't make sense to incorporate patients into a study that do not have the genomic predisposition for response, and particularly not if they have a genomic predisposition for adverse impact. NGS biomarkers can make a big difference, and we're going to talk about 2 cases here. One is an NGS biomarker in arteriosclerosis, the hardening of the fatty deposits in the arteries, and how 5 functional alleles were discovered that invalidated a therapeutic target. Unfortunately, as you will see, a little bit too late to save this particular pharma company $250 million. The other are NGS biomarkers in immuno-oncology. Immuno-oncology is also emerging again as an area where the genomic signatures for the treatment are becoming more refined, and our targets are becoming more refined. And we're going to look at how whole exome analysis of tumor mutation burden and, interestingly enough, microbiome shotgun sequencing of the gut microbes are leading to biomarkers for immuno-oncology. Let's take a look at that first case. This is a drug called daraplatinib. And again, this is things you really wish you knew before going into a phase 3. This is a promising drug developed by GSK to control arteriosclerosis, and it worked by targeting a lipoprotein-associated phospholipase A2, also known as LPPLA2. It failed spectacularly in phase 3 due to efficacy, resulting in a $246 million write-down. Should be a big M in there, sorry. And it was cited as the #2 clinical trial failure of 2013, not an award that you really want to stand up for. Coincidentally, Baylor and DNAnexus were performing genome-wide meta-analysis testing on whether this LPPLA2 is actually relevant to coronary heart disease. And the genomic analysis in combination with the clinical data for the participants revealed that none of a series of LP2PLA2 lowering alleles was related to heart disease. So what happened here is we found lots and lots of participants who had alleles, who had mutations, variants, that would lower the impact of the LPPLA2, but we did not see that related to heart disease risk. These folks still were showing signs of the heart disease. This essentially indicated that this particular target was not a causal risk factor for this heart disease, and as such was not a drug target. Again, you really would like to know this in advance, and by having a cohort of clinically sequenced participants, one could associate their genomic makeup with their predisposition towards this heart disease, and it would have been discovered that this phase 3 should not have even been entered into. For immuno-oncology, we see a couple of areas where you can predict therapy efficacy. And again, you do not want to be getting participants that are inherently genomically not going to be responsive to treatment. This is a treatment for lung cancer, and it is using a mutation burden. In this case, it's non-synonymous mutation burden in tumors, and this mutation burden has been distinctly associated with improved response and durable clinical benefit around progression-free survival with these patients with non-small cell lung cancer. And these patients are being treated with a drug called pembrolizumab. I have a hard time with these drug names. This is an antibody that targets programmed cell death 1, the PD-1 target. Incorporating analysis of the tumor burden here, you can see that the higher neoantigen burden is very strongly associated with longer periods of progression-free outcomes. Similarly, the influences of IO therapy efficacy can be discerned using metagenomic shotgun sequencing of the participant's gut microbe. What we're seeing here is metagenomic species counts on the left, and on the right is the microbe gene counts, and there's a distinct pattern of difference between the makeup of the microbes in these participants, again leading to progression-free survivals of greater than 6 months and less than 6 months. So what does all this mean? It means that if we incorporate NGS into our clinical trial design, we have a much higher chance of putting together a participant cohort that will respond effectively and not show adverse interactions. And what does this mean for IT? Well, again, there are molecular considerations, there are clinical considerations, and there's a regulatory overlay on all this. And this will describe how DNAnexus has over the years looked at this problem space and arrived at a clinical trial solution that enables NGS to be incorporated and optimize and de-risk clinical trials. This is the heart of a cloud-based clinical trial solution. The box in the bottom, the clinical trial solution running on the Azure cloud on DNAnexus, provides full integration and audit capabilities for good lab practices as required for clinical trials and for adherence to the Code of Federal Regulations 21, Parts 11, which have to do with storage and handling of electronic data records, and Parts H20, which has to do with validating quality management system. One of the things that one needs in an IT environment for clinical trials is to have a quality management system. It's not enough to have just an information security management system, which is suitable for delivering clinical diagnostics, but additionally, you need to have a quality management system showing that quality is being measured, tracked, and updated continuously. A study manager as part of the solution enables many different sites, many different studies, different sequencing labs, and all to be integrated and managed easily with all the genomic data handling, the incorporation of clinical data, and the outcomes clearly managed on a study-by-study basis. Workflow publication is also necessary to ensure that validated production pipelines are the only ones being executed on the inputs coming in from these sequencing labs. Computer system validation is one of the more onerous aspects of this regulatory landscape. And really what that means is, from an IT perspective, any computer system that you're injecting into the clinical trials decision process that you have built needs to be validated all the way from requirement specification through to production delivery using a requirements trace matrix. This can be a really heavy lift from an IT perspective, and by incorporating the computer system validation centrally into the DNAnexus cloud-based clinical trial solution on Azure, our customers can focus strictly on validating their bioinformatic pipeline and not worry about doing installation quality, operation quality, performance quality, and traceability on all the computer systems that they have that make up the clinical trials decision process. The insight from NGS, getting it accurately, reliably, and efficiently requires world-class IT configurations as well as scientific expertise, including human genome mapping and alignment, variant calling for single nucleotide polymorphisms, copy number variation, insertions and deletions, structural variation, transcriptomics, human microbiome strains analysis, and characterization. There is a secondary analysis solution that also runs on DNAnexus and Azure, which essentially feeds the genomic information as it comes out from the sequencing labs into the platform in order to have genomic information to relate to clinical information to seek insights. Janssen has deployed this for a inflammatory bowel disease test. This is a really troubling set of symptoms. I'm sure everybody here on the call has folks that have dealt with Crohn's disease and it's a very lifestyle impacting set of diseases. And what Janssen has been working on is clinical trials around a microbiome-based treatment, actually a live biotherapeutic. The Janssen pipeline designers work on platform to develop and harden their pipelines that will process and understand the shotgun sequenced microbiome in order to determine the strains and the levels of strains and genomes, microbiome genomes that are in the sample. By working on a common platform for both development and production, the pipeline designers, again, do not have to lift and shift between various regulatory environments. They can operate in a development environment without the onerous computer system validation, but still have all the change control, version management, and all of the other development features to enable effective development and effective transition, efficient transition from a development environment into a hardened production environment. The bioinformaticians deploy and ultimately harden their pipelines for use in the actual clinical trial solution. Validated applications are managed through the workflow publication system, and participants' DNA samples are ingested through the study sites by study physicians and handled through central labs and multiple sequencing labs that upload the raw sequencing data, generally FASTQ files, into the DNAnexus platform, where the mapping, alignment, and variant calling takes place. The service providers can track their status of their sample uploads and the sample processing. The study managers and study clinicians can handle any errors and restarts that might be necessary in processing the genomic samples. And by using the API around the clinical trial solution, we interact with the clinical systems of record, namely the electronic data capture system, where any reports that are being generated will be deposited, and the clinical trials management system, which is orchestrating the entire clinical trial. This is analogous to interacting with a laboratory information system in the case of a clinical diagnostic. These are the systems of record, but they are not built to store 200 megabytes or a terabyte of tumor normal data or proband parent trio data. So the clinical trial solution essentially extends the system of record to store the larger data, high throughput data files, and to be able to perform the higher intensity analytics that are required for these high throughput data files. So what does this mean from an IT perspective? What do you have to deliver in order to adhere to all of the information security, quality, and compliance management to be able to operate a system like this in a manner that is reliable, highly available, and has a strong confidentiality, integrity, and availability profile, which is required for clinical diagnostics and for clinical trials. DNAnexus essentially has worked on top of the Azure Cloud Service Provider, which itself has many certifications, but that's not quite enough. And this is a really key point. There is a lot of movement, I wouldn't say movement, but there's a lot of fallback at times to what I'd call a DIY on cloud perspective. This viewpoint can be espoused, I think, in a manner that it tries to hew towards cost savings, but really ends up being penny wise, pound foolish. Because just because you have an infrastructure as a service upon which you can get a business associates agreement under HIPAA, it's certified independently for ISO 27001 information security management, similarly for FedRAMP and SOC 2, and certified for ISO 9001 quality management. What you have are building blocks, and certainly you can build a solution, but you don't just build it, you have to build it and operate it in a continually secure, compliant manner. And that presents a considerable amount of risk and a considerable amount of cost and effort and time. There's the time it takes to build all of these things and the time and energy it required to run these in a secure and compliant manner. And this is where building an IT system with all these considerations in mind requires thinking about these boxes that sit inside the DNAnexus portion of this diagram. So this layer cake is essentially reflecting how you can take underlying Azure compliance, combine it with DNAnexus features and DNAnexus certified compliance, in order to deliver compliant services such as the clinical trial solution or clinical diagnostic solutions that are going to adhere to local regulatory requirements for data handling, like the EU GDPR, to clinical diagnostic requirements and protected health information data handling, for federal requirements, such as the FDA in wanting to receive NGS-based evidence for sponsors submitting NGS-based drugs and devices for review, DB-GAP for wanting to incorporate public data into other types of services, and of course, relative to clinical trials, being able to adhere to GXP and the relevant federal code of regulations. Let's take a look at some of these features. First, we really have to run this, as I said, and operations is not just the day-to-day management of the cloud-based infrastructure, but it is the ongoing application, engineering, and operation of the information security, quality management systems, and ensuring that the regulatory regimes are addressed that we've mentioned previously. We provide features for data access control on top of our cloud-based system. This goes beyond just having access control lists for various blob store buckets or other things like that, which are a fairly primitive way to address things. If you think about it, you really don't want a bioinformatician who would like to share data with a colleague dealing with blob store access control lists. They really would like to be dealing with a higher level of abstraction for how they control access to their data. The other attribute of this IT infrastructure is to ensure that data is immutable. As you can imagine, you do not want data of record, particularly in pipelines for clinical trials and diagnostics that really need to be reproducible. You really can't have the data moving around underneath you. So you need a platform that once the data is deposited, it cannot be changed. This is also tied to unique data identification through the use of GUIDs, so that a rose is a rose is a rose by any name, but that data is uniquely identified and cannot be changed, even if it's shared, copied virtually. It essentially is a piece of data that you can count on as being the same data that was uploaded. Complete encryption, both in and out of this IT infrastructure and within this IT infrastructure, is mandatory. Ideally, the data really is unencrypted only when it's being analyzed, and ideally, again, only in RAM of worker nodes during the time of analysis, after which any unencrypted data is discarded. Monitoring and threat management is a crucial aspect for any of these systems. Again, this is an area where the DIY doesn't really buy you anything. You really do have to keep up on top of operating system, application level, web level threats, viruses, and other types of security challenges. And that is a very full-time job. Logging and auditing are also key features to support clinical compliance, both in a clinical trials and a diagnostic setting. Reproducibility and demonstration of controls through the logging and auditing reports are required in order to be able to satisfy regulators like the FDA. Chain of provenance is conferred essentially by the combination of immutable data and application versioning and complete logging and auditing. Any piece of derived data that's being used for a clinical trial decision process or to inform a diagnostic call, we need to know where that derived data came from. And by having a data chain of provenance, I can take any piece of derived data, look upstream to understand the exact version of analysis code that was run, the input files, the reference genome, the configurations that were used, the configuration parameters used for the applications, and all of the logging from the actual execution of the application across all the various numbers of worker nodes that it may have fanned out to. This enables a very definitive chain of provenance tracking for derived data and supports demonstrable reproducibility of results in a clinical trial and a clinical diagnostic setting. In order to do collaborative analysis and to have data access controls, we need identity management and a way to link the identity management to access controls. You need a platform that'll federate with external directories like Active Directory and can manage groups and users inside the system in order to enable really convenient, flexible, yet reliable collaboration. Collaboration sits on top of security. There's no way to have reasonable collaboration without solid security, identity management, and data access controls. I mentioned previously this computer system validation. All of these systems that are used to inform a clinical trial decision need to be run on computer systems that have gone through formal installation quality, operation quality, and performance quality testing, and have been demonstrated to show that the specification for this system has been validated through requirements, development, testing, and ultimately into production as it's incorporated into the clinical trial. This is one of the most heavy lifts that all the amount of DIY really won't solve. Even doing it DIY, you have to go through this onerous process. And by having a central system where this is all handled internally, it frees up the clinical trial designers and the clinical diagnostics designers and the developers to focus only on validating bioinformatic pipelines and handling of molecular and clinical data. Metadata integration is also very important. The system has to be able to ingest metadata from upstream systems, like a clinical trials management system or a laboratory information management system, to hold and carry that metadata along with the participant sample and to propagate that data with the report result or whatever fields of data results are being transmitted out of the clinical trial or clinical diagnostic system. So building on top of this foundation, the secure compliant foundation at the very bottom, where we're essentially building on these features, operations, information security, quality, and compliance that I just alluded to previously, all on top of the Azure features. We basically use some fairly straightforward Azure features, virtual machines, file and blob store, the file store, generally for transient storage on the virtual machines during the time of analysis. The blob store being highly resilient and much lower cost per gigabyte than the file store is used as the system of record, the storage system of record. And data is marshaled from the blob store to file store on active VMs when we want to analyze it. We do take advantage of machine learning fabrics, the excellent Azure machine learning capabilities, and of course, all of the compliance features and certifications that Azure provides. So building on top of what I've mentioned in the previous slide, we have a critical construct called the project. The project is really the governance boundary. And again, as I said before, you don't want a bioinformatician dealing with access control lists on Azure blob store buckets. You don't want them dealing with how to spin up secure virtual private clouds and putting the virtual machines in there. That is not the language of bioinformaticians. The language of bioinformaticians is workflows, applications, and data. We provide a mechanism to use containers, Docker containers, which allow both import and export of apps into this platform. And we use common workflow. These are essentially languages that serialize workflows to describe complex workflows, basically directed acyclical graphs using a serialized textual language. Two of these in place are the Common Workflow Language and the Workflow Definition Language from the Global Alliance for Genomic Health. I mentioned users and groups. These are all tied into the project structure through project access permissions. And by having the users and groups able to share projects, it makes it very straightforward for researchers, clinicians, study designers, clinical diagnostic designs to create a project structure that enables partners, internal developers, and others to collaborate around the workflows and apps and data contained in projects. Additionally, the world is not just a batch world of pipelines and file data. In order to get signal out of the combination of genomic data and clinical data, we need structured data stores such as Apache Spark. We need analytics and visualization and business intelligence support through database connectors. This is done using R, Jupyter notebooks, and again, a database connector to enable tools like Spotfire and Tableau and other business intelligence systems to do cohort browsing, associative analysis, and machine learning to tease apart the relationship between genomic data and phenotypic expression, generally for the purpose of discovering biomarkers. So this project construct is a way to take the underlying primitive objects provided by the cloud service provider, by Azure, and wrap them up into something much more user-friendly and bioinformatician and clinical trial and clinical diagnostic designer. The way to think about this is I can take these projects and share them with read-only access, upload capability, a variety of different permissions, but that project encompasses all of the batch data, file data, workflows, analytics, the Apache Spark cluster, any of the Jupyter notebooks that have been used to present analysis and visualizations, and to be able to readily share that with other groups and users in a very secure and compliant manner. The next very important aspect of any IT system that needs to deliver in this environment is that it must be accessed via API. This cannot be a case where bioinformaticians are directly accessing objects in file stores or blob stores. This API essentially enforces the compliance boundary because there's no way to get at information or to get at functionality without going through the controlled, secured, authenticated API interface. Any web interface to the system should be built on top of the API exclusively, no backdoors. The command line interface, which is the familiar place for bioinformaticians to work, again, sits on top of the API, much like the software development kits, again, for bioinformaticians who want to implement Python and Java pipelines. Critically, the API supports upstream and downstream integration. This integration is through, basically you can have a LIMS system, which is a master for a clinical diagnostic or feeds in the samples for a clinical trial participant genomic analysis. And that LIMS system will interact with the API to generate the genomic results, generate the reports and interpreted data, and essentially the clinical reports, and to integrate this with downstream systems such as the electronic data capture, or in the case of diagnostics, even back up to the LIMS system. On top of this API, the solutions for clinical trials, clinical diagnostics are built with accompanying solutions for secondary analysis, which essentially will be used to feed the genomic and molecular data into the clinical trials and clinical diagnostic solutions. And very importantly, the translational informatics solutions, which will be used to relate genomic features to phenotypic expression in order to discover genomic biomarkers. The secondary analysis solution I'll touch on briefly. This is where you're going to see batch analysis with workflow support, applications for DNA and RNA-seq seq analysis, microbiome analysis applications, reference data libraries, and workflows. Again, the secondary analysis portion of the platform will feed the clinical trials. Again, all of this stuff operates through projects where you have a project which is related to a given study. That study and that project has various contributors and collaborators, whether they're the sequencing service provider, the study physician, the study manager, all of that, and with the accompanying secondary analysis workflows and data will reside in that project. The translational informatics solution is really about integrating the genomics and other omics data, whether it's imaging data, proteomic data, computational chemistry data, with trial results, clinical data from EHR, or from the actual clinical study results and real-world evidence, bringing that all to bear in combination with the genomic data to perform statistical analysis and machine learning to derive insights into the relationship between the genomic data and the phenotypic expression. This would lend itself to visualizations where you can visualize these relationships, and cohort browsers where you can slice and dice the participant cohorts into various pieces based on their genomic makeup, their demographic makeup, and their clinical expression. The compliance continuum reflects the fact here that with a single platform, you can do all of your discovery of biomarkers. And again, in early development, you're operating in a research context without a lot of onerous regulation, yet you still need version control, immutable data, all these other things to help support reproducible research, the outcome of which are discovered biomarkers that are detailed, highly characterized. Once these biomarkers are discovered, you want to move them into a clinical trial environment. And that can be done by simply taking these same applications and reference data and moving it and qualifying these applications and creating validated pipelines all on the same platform. And again, taking the raw sequencing data and metadata, we're now generating signature calls that can be used to segment and stratify participants, and very importantly, to refine inclusion and exclusion criteria to enhance the levels of responders and diminish the level of adverse impact on subsequent phases of the trial. One of the trial design attributes here is the incorporation of this discovery process into a phase 1 with the intention of using the result in a phase 2 for participant stratification and inclusion and exclusion criteria. This also requires this decision process to be operated under the clinical trial regulations, essentially the GLP stack. Pardon me. Once a diagnostic moves to post-market, the same application of biomarkers, either to determine whether a patient is right for a treatment or to track the progress of treatment, those pipelines can be moved straight into a clinical diagnostic context. And here's an example of a clinical diagnostic solution where a non-invasive prenatal test is being delivered in a similar manner as a clinical trial. However, rather than having clinical trial sites and sequencing service labs that are sequencing participants interfacing with the cloud-based platform, these are diagnostic test lab partners. And in this case, Natera deploys in a manner similar to bioinformaticians developing a clinical trial pipeline. They deploy their diagnostic test pipeline, test it, stress test it, validate it, and then push it to production where it's available for diagnostic test labs to deliver this non-invasive prenatal test anywhere in the world. Essentially, you've got a distributed diagnostic test anywhere, deliver the report anywhere using a single version, proprietarily protected pipeline in the cloud. This whole environment from an IT perspective needs to adhere to CAP/CLIA requirements, essentially treating this informatics and IT environment as an external lab with respect to CAP and CLIA regulations. The genomic data flow on DNAnexus comes about through post-sequencing. We've talked about the processing of the genomic data, the BAMs, the variant call files, the microbiome files that are the output of the scientific pipelines, the combination of multi-omics like transcriptome, RNA-seq, microbiome, and epigenetics, methylation, also into the genomic data, the incorporation of phenotypic data from EHR, imaging, and study data, all in generally standard formats like the OMOP clinical data model or the study data tabulation model from the clinical data interchange standards, getting all of this stuff wrapped up to be able to use the translational informatics tools to enhance the phenotypic data, to enhance the genotype data and the phenotypic data to end up with derived geno and pheno data and to feed these into statistical association and machine learning processing. This is done with a translational informatics suite. And again, if you recall, the Spark, the R, the business intelligence, and the Jupyter Notebooks aspects of the project resources, those project resources are used to feed a translational informatics solution, which ingests all of the genomic and other omic data with clinical trial data, EHR, and real-world evidence data into appropriate data stores. A variant warehouse looks a lot different than a phenotype warehouse. Variants definitely do not lend themselves to relational databases, whereas phenotypic data stores are inherently relational based. The refinement of the genotype and phenotype data through things like gene expression analysis, phenotypical binning. You might not want 14 different definitions for cancer of the mouth. You might want to roll that into a single one. You might want to take a genomic signature, which involves multiple genes, and combine that into a signal. But you end up with derived, statistically refined genotype and phenotype data, which get fed into interactive and visual analysis and rich analytic applications. This all leads to biomarker identification, ability to de-risk clinical trials based on safety and efficacy, and most importantly, to know things in advance, much like the drug target we looked at before, which should never have entered a phase 3 if we had that genomic information well understood. To understand indications and evidence generation for clinical trials, and to support pharmas in their drug research and targeting new drug discoveries. Excuse me. Very quick presentation of DNAnexus. I want to save some time for questions. We're essentially two pieces. One is the platform as a service, which we've talked about, which is how you wrap up IT, essentially cloud-based Azure resources, into a package that's easily managed from a regulatory and compliance perspective and presents bioinformaticians, study managers, clinicians with an interface that makes sense to them, not a bare infrastructure as a service cloud interface. And this is combined with our scientific services, bioinformatics, integration, application qualification. We have about 100 people, 5 offices worldwide with headquarters in San Francisco Bay Area. 15 out of the 20 global pharmas use us. The FDA has deployed Precision FDA on top of our IT infrastructure, the DNAnexus Platform, and our platform resides on top of multiple regions running on Microsoft Azure. We serve essentially 3 markets. We are in the pharma and biotech sector. Many examples of that. We operate with government entities like the FDA and participate in large NIH-funded projects like CHARGE, ENCODE, and eMERGE. And we work with some of the major sequencing centers, both academic and commercial, like the Baylor Human Genome Sequencing Center and the Garvan Institute, which provides research and clinical exome and whole genome. In clinical and healthcare, we operate clinical diagnostics, many different cases, non-invasive prenatal tests, rapid diagnosis for neonatal intensive care children with undiagnosed diseases, oncology, and pediatric cancer, and of course, just plain old clinical exome and clinical genome sequencing, generally for carrier screening. I'd like to make a announcement for our conference with next week. We'll be working with, we'll be in the Microsoft booth, and we would look forward to your attendance and your contact out at the HIMSS Conference 2018 in Las Vegas next week. I think that's it, and we have time for any questions and answers that might have come up.
Thanks, Omar, for that. So the Q&A section and the chat box is open to take any questions that you have. And we have a couple of minutes to go. So the first question for you, Omar, is what do you think is the most impactful, transformative insight to making decisions in healthcare coming from NGS?
Clearly, biomarkers are probably the heart of it, mainly because if you have a biomarker, you're 5 steps closer to having a drug. I've heard colleagues in pharma say that without a biomarker, you're really not going to have a drug. And seeing the level of essentially the 3x chance of getting out through a successful phase 3 that's enabled biomarker, I'd say that's really the important part. And in order to do that, you really need to be able to combine the genomic information, the phenotypic information, any other omics, proteomics, transcriptomics, anything needs to be brought to bear to essentially discover. As I said earlier, no biomarker left behind, I think, is a really critical focus.
Great, thank you. So we have another one. How can IT embrace NGS on its way to universal access, and how do we accelerate or leverage its value?
I think the regulatory and compliance is probably the first sort of point of entry. Without having appropriate regulatory and compliance control, it is very difficult to see how NGS can advance from a state of privacy, protected health information. NGS data is unique, even in clinical data, in that when you move more towards whole genome and even whole exome, the concept of de-identification becomes more problematic, because you can think of a variant file for a given individual as being akin to a fingerprint. So having the regulatory and compliance aspects nailed down is very important, and having it nailed down in a way that's cost effective and efficient, it doesn't require a major amount of distraction, investment, IT staffing, I think is very important. You have to make regulatory compliance easy. I think that's a very key aspect.
Great, thank you. And just one more. What do you anticipate will be the biggest needs and or challenges regarding big data in hospitals?
I think the scale of the data is just going to be similar to clinical trial systems. It just doesn't align with hospital systems. I think that hospitals will need to embrace external services appropriately secured, likely interfaced through SmartFire APIs, to be able to integrate things like 1 terabyte tumor normal or proband trio data samples into clinical care. Ultimately, I'd say at the hospital site, you're going to see things like large variant catalogs that will be used to inform clinical care, but the raw data files that will be used to generate those variants, they need to be maintained and managed as well. And I think that's the bigger challenge for a hospital system, is to integrate larger scale data and, of course, the analytics scale that comes with it, the hundreds of cores or thousands of cores that may be required to analyze that proband trio.
Great, thank you, Omar. And with that, unless we have any other last minute questions coming in from any of the attendees, I would like to say thank you, Omar, for taking time out of your day to present this webinar. And thank you everyone for attending our webinar as well.
My pleasure, and thank you for your attendance. Really appreciate it. Hope to see you at HIMSS.